Hello everyone, I'm Charlotte from MDPI and I will be your host for today. Welcome to the second live session of the first International Electronic Conference on Biological Diversity, Ecology and Evolution. This conference has been organised by MDPI and the Diversity Journal. I wanted to take this opportunity to thank our chair, session chair and conference committee for making this conference possible, and also the speakers and participants for joining us today. This conference is now open for discussion. Until the 31st of March, you can log in with your SciForum account and read and comment all submissions presented in the conference. As you know, live sessions are being organised throughout the conference, so please check the conference website for more information. Registration for the next two live sessions is still open. I would also like to remind everyone that this live session is being recorded and that the recording will be available on the conference webpage as soon as possible. The webinar is also being live streamed on MDPI's YouTube channel, so hello to those of us watching on there. Today, we will have a session dedicated to biodiversity and conservation with a panel of 5 excellent speakers. The whole session is chaired by our session chair, Professor Paolo Giordani, from the Department of Pharmacy at the University of Genova in Italy. Thank you, Paolo, for accepting to chair this live session. We will have a time for questions after each talk, so please type your question in the Q&A box at any time during the session. Please make sure to add the name of the person to whom you're addressing your question. That's it from me. I now hand over the microphone to Professor Giordani and wish you a pleasant webinar.
Thank you, Charlotte. Thank you so much. Welcome everybody to this session. We will have a considerable set of presentation ranging from several aspects of biodiversity. So we will speak about what is going to happen in the very next future to plants. We will have a very interesting presentation by Professor Gouyon on the effect of pesticides on biodiversity. And again, another presentation on spider webs by Dr. Gregorič. And a further contribution by Dr. Wilson Frantine-Silva about diploid males in a suitable environment. And finally, a further contribution by Dr. Maria Panitsa on plant species diversity in biogeography in Greece. So I will introduce a little better each speaker. Now I will leave the floor to Dr. Lucia Varaldo from the University of Genova. She will present a contribution dealing with can species distributions models help to design conservation strategies for narrow-ranged species under climate change, a case study from Santolina Genus. So please, Lucia, the floor is yours.
Hello to everyone. I am Lucia Varaldo from University of Genoa. And okay, let's start. So climate change is increasingly affecting the distribution of the species, in particular those who have a narrow distributional range. To cope with the climate change, the species could adapt or shift their distribution. But considering species with narrow distributional range, these two options could be conditioned by the great intrinsic vulnerability. In fact, low population size, low ecological tolerance, and low genetic richness makes adaptation difficult, and poor dispersal capability prevents shifting. So in situ conservation of natural habitats of the species may be inadequate to reduce the loss population. And therefore assisted colonization may be necessary to assure species survival, moving individuals into new suitable areas. We selected as study area Mediterranean basin because of its richness in biodiversity, especially in species with narrow range. And we have additionally to consider that it's one of the regions most vulnerable to global change. We selected 3 species of Santolina that belonging to Santolina genus. That is a genus widely distributed in Western Mediterranean basin, and that includes many endemic species with narrow distributional range. We selected Santolina decumbens, Santolina ligustica, and Santolina pinnata. The aim of this study is to understand how future climate change could influence this species of Santolina, and how useful could be assisted colonization to assure species survival. The 3 species are endemic in the western part of Mediterranean basin, and all they have a relatively high number of occurrences in area. Santolina decumbens is a French endemic species in the southern France, in Provence, Marseille, and its altitudinal range is included between the coastal and the mountainous belt. Santolina ligustica, it's an endemic plant that grows in Liguria, my region in Italy. And its altitudinal range is included between the coastal and the hilly belt. And the last species is Santolina pinnata that grows in Tuscany on the Apuan Alps between the hilly and the mountainous belt. To study these species, we use the species distribution models technique, that is a statistical tool. The models are a simplified representation of reality, obtained correlating climatic variables and observed distribution. They return an habitat suitability maps, and this technique is commonly used to predict species distribution, but are still rarely used in translocation planning. To build our models, the occurrences obtained from local database and from the project Plants 2.0, that is a project between Italian university, including also University of Genoa. And we selected only occurrences tracked using GPS, so only precise occurrences. And we obtain climatic variables obtained from CELSA climate database. We downloaded 19 bioclimatic variables, so for current and future time. And we selected among those available to the most optimistic scenario and the most pessimistic one, considering for each of them 5 global circulation models. We have to remember that we are talking about narrow distributional range. So to take into account microclimatic condition, we downscaled at 50-meter spatial resolution the bioclimatic variables. And then we used the first 2 axes of environmental principal component analysis, PCA, as environmental variables. To build our models, we used, and then we obtain maps also for current and for optimistic and pessimistic futures. We did ensembling, calculating average values, and then we transform these maps in maps, as you can see in the picture. To assess of the position of possible areas for translocation in relationship to species distribution, we consider 3 different areas. The distributional range, calculated as convex soil around occurrences, an area 5 kilometers larger than larger than this distributional range, that is the yellow line in the picture, and 1 kilometer buffer around occurrences, that is the red circle in the picture, in the presentation. The 3 species to a range loss higher in the pessimistic scenario, but we can detect some difference in the species. In fact, Santolina decumbens and Santolina pinnata, under optimistic scenario, show an expansion of their distribution. Differently, Santolina ligustica show a moderate contraction. Under pessimistic scenario, Santolina decumbens show a moderate contraction, and Santolina pinnata and Santolina ligustica show a total contraction, losing all their distribution. These results are consideration to be made. Santolina decumbens and Santolina pinnata expand under a moderate global warming in line with previous observation in other Mediterranean Thermophilus species. The ranged contraction detected in all the species under the pessimistic scenario underline the importance to reduce the CO2 emissions to assure biodiversity conservation. And Santolina pinnata and Santolina ligustica have a narrow altitudinal range, and they are more threatened than Santolina decumbens. This is in line with the idea that species sensitivity to climate change is affected by niche properties. If we consider the three different areas, we can see different in the species. In fact, Santolina decumbens under both scenarios will have future suitable areas close to occurrences. Differently, the other two species, Santolina ligustica and Santolina pinnata, will have future suitable areas far from occurrences. And in the under pessimistic scenario, they will have a range loss higher than a range gain. The ability of the species to cope with the climate change is related with the possibility to reach new suitable area that is in turn related also with the location of the suitable areas and the dispersal capability. All the three species of Santolina have a poor dispersal capability, having seeds with low efficient plume. Santolina decumbens is the species with the widest altitudinal range, and so this species could shift its distributional range. And that's why it's needed. For this species, in situ conservation would be enough to assure species survival. The other two species have a narrow altitudinal range, and they would be able to adjust their geographic distribution appropriately, and it would be more affected by the climate change. In Santolinata, persistence of suitable areas close to current distribution would allow in situ conservation, but also translocation may assure species survival. Differently, Santolina ligustica, for which future suitable area would be outside of current distributional range, it would be necessary to carry out a system colonization. I go fast. Thank you.
Thank you. Thank you so much, Lucia, for remaining within the time. So we have saved time for question. We already have one. I'm going to read it for you by, sorry for the pronunciation, Anatoly Kapugin. I hope to have pronounced fairly. And well, I summarize a little bit. Anatoly asked you if, did you provide for these species of Santolina the assessment of conservation status according to the IUCN red list categories and criteria? If you have already done it, or if you plan to do it.
Yes. So there is no study. The three species are not inserted in the IUCN list. There is no information about them because they are endemic species, but they aren't protected, so they are not in the IUCN list. Santolina ligustica is smaller than the other, and I think that it's important more information about these species to planning management, to planning conservation plan.
Okay, thank you. Anatoly, if you want to add it directly, you are kindly invited to do it. Otherwise, I summarize. So don't, I mean, feel free to contribute. Yes, another note by our colleague. He believes that the determination of potential species distribution is a good supplement to the traditional assessment of IUCN. So meaning yes, that in a near future would be vital to include this kind of approach into the assessment protocol, basically.
I agree, totally agree.
Okay. Any other question from the floor? I have one for you. Did you speak about the niche characteristic for explaining the differences in the range shift of the free species? I got it correctly. But what about the, well, for instance, some biological traits of the free species could be somehow related to these different ranges, or they share more or less the same traits?
No. They are the same, belonging to the same genus, but they grow on different soil. So maybe also the possibility to have this particular soil around the occurrences could influence, but in this study we didn't consider this. For example, Santolina decumbens, that has the widest altitudinal range, grow on basic soil. But Santolina ligustica grows on ophiolitic soil that are more rare in around the occurrences. So maybe that's why the future suitable areas wouldn't be near to current distributional range. So surely this condition would influence.
Okay. So the trade-off between their capability to disperse and the occurrence of suitable substrate could be another limitation for their expansion.
Yes.
Well, great. Thank you so much, Lucia.
Thank you.
And we are. Is there any further question? I think we have a couple of minutes still. Claudia Brunetti, okay, I read it for you. She asking you if you think that the citizen science projects can be an helpful tool in monitoring the potential species distribution and to expand the knowledge about this genus.
Maybe yes. I think yes. With the other university, with the other Italian university, we are trying to clarify this genus because it's really complex. And so we are making some genetic analysis. So on DNA or on seeds, we are taking into account many characteristics to try to understand better this genus and reorder, maybe. We don't know yet.
Okay. Many contributions for your speak from Professor Michael Vink. He's asking, how did your species survive during the last glaciations? If you could tell something about this.
Sincerely, I don't know. I have no information about this, sincerely.
Maybe, I don't know if the area they are currently occupying were covered by the ice? Actually, I think no, maybe. For the 3 Italian species, for sure no, but...
No, it's widely distributed in the Mediterranean basin, and there are many species, but they live in the coastal belt in general. Some of Spain, in Spain, some species are more higher, the altitudinal, but I don't know sincerely during the last glaciation what happened.
Thank you so much, Lucia. I think we can move to the next speaker, and I introduce you Professor Pierre-Henri Gouyon from the Museum of Natural History of Paris, I guess, and he's going to give an interesting contribution about biodiversity and pesticides, why we are wrong. I think I read the abstract. I'm very curious about the contribution of Professor Gouyon. So thank you so much for coming, and the floor is yours.
Thank you very much, and thank you for being there. So through a bit more than 3 years ago, more than 15,000 scientists from almost 200 countries issued a warning to humanity. This came from the Union of Concerned Scientists, who had already actually warned humanity in 1992 about the problems of the planet. 1992 was the year of the Rio Earth Summit, and the Union of Concerned Scientists wrote this text, and it had absolutely no impact. It had no impact because another text was published exactly at the same time, saying approximately the reverse, while trying to show concern too. It was called the Heidelberg Appeal. It was signed by 4,000 people, including 62 Nobel Prizes. And in this text, which you can easily find on the web, it is said that people want to make serious things about the Earth, our planet, and so on. But actually, the message which is sent is absolutely against environmentalists, against ecology. They say that we are, however, worried at the dawn of the 21st century at the emergence of an irrational ideology, this is ecology, which is opposed to scientific and industrial progress and impedes economic and social development. We do, however, forewarn the authorities in charge of our planet destiny against decisions which are supported by pseudo-scientific arguments or false and non-relevant data. So see, this text pretended being in favor of the defense of the planet, but actually it really attacked environmentalists and defended industry. And indeed, a lot of books, for instance, have criticized ecology and environmental environmental concerns, calling them austerity ecology, and defending growth, process industry and stuff. So we have a problem of the vision of is ecology against the development, and also the question is how do scientists decide on which side they go? So actually, we now know that the Heidelberg Appeal was a scam perpetrated by the asbestos and tobacco industry, and it was funded and controlled by a coalition which later included coal, oil, and energy. And this is the basis of what sociologists have now called merchants of doubt, people who are in charge of rejecting the realities which are not fine for certain industries. The first guy who showed these problems was named Scott Cutlip, and in 1994 he wrote a book called The Unseen Power, showing that there was a very strong possibility of action of what is now called social engineering. And the way he did that, the way this was done, started in December 15, 1953. And actually what happened on that day is that the major tobacco companies met John Hill, who was the head of the public relation company, and he explained them that the problem with tobacco, the health risks would be demonstrated, and if they just denied them, it would not work. That the good strategy was to create a major scientific controversy to fund research so that you can prove that the problem is multifactorial and that more research is needed. So this is a very important point. If you see that a problem is multifactorial and that more research is needed, well, this is almost always the case. But the problem is, if this is very much insisted on and then nothing emerges, then it's very likely that some merchants of doubt are at work. Concerning tobacco, this has been studied in detail by Robert Proctor, who wrote a book about that, The Golden Holocaust. And he, in particular, he cites a document written by the WHO, which showed how much they had been infiltrated, how much they had been cheated by the tobacco system. This text is 200 pages long, and it shows how the tobacco industry has really cheated with their system by establishing appropriate relationships, including political and financial power to influence, using other UN agencies, including the Food and Agriculture Organization. This will be useful for us later, etc., etc. There's a list of what they did to them. And the tobacco system paid very great scientists Nobel Prizes, great physicists like Fred Singer, great scientists like Frederick Seitz. All these guys became the team. And why did they do that? Why did great scientists go into selling doubts and assessing things which were not true? Well, actually, well, first they received money, certainly, but also there is an ideological dimension. These guys did not like communists, you know, that was in the middle, well, in the second half of the 20th century. And they were explained that ecologists were like watermelons, you know, green on the outside and red on the inside. So the alliance of ideology and money made that these scientists went into this merchant of doubt things. And after the tobacco system, well, the next users of this system were the oil companies who provoked deniers for climate change. Well, you've all heard about that, so I won't go too far. But you know, this is the same system and the same way of denying the reality by pretending, doing some research, and again because the green people are seen as enemies of progress. Now, let's come to our subject today. In this context, the first person who really alerted us against pesticides was Rachel Carson in the 60s. And she proposed the idea that actually we were going into a silent world because birds would disappear because of the disappearance of insects. She succeeded in having DDT forbidden, but unfortunately, hundreds of other molecules replaced the DDT. And a new book was published in France this year about this same question, and I want to show you some of the results which are shown in this book. The book starts with a problem which is the colony collapse disorder of bees. You know that colonies of bees disappear very, very heavily. In some places, more than 90% of the colonies go extinct. They go extinct because the workers do not come back, actually, and then the queen remains alone with a few bees, and then the colony collapses. There's been a number of studies about that. And if you look at the American Environmental Agency, what they tell you, and you can see that on the web, and if you look on the web at what is said about this, it's very interesting because oops, sorry. what you will find usually is that there have been many theories about the cause of CCD. The first cause which is given is very often the mite, the varroa. The varroa is this little pest, this little animal which sucks the bee, you know. And then there are viruses, and there is a gut parasite, nosema. Nosema, I show you here. Well, you see an advertisement about hives and nosema. And pesticides poisoning through exposure to pesticides applied to crops and or for invasive insects or mite control. So actually, the controlling the varroa can be deleterious for bees. Stress bees due to management practices because there are transportations or changes of the habitat, inadequate forage, poor nutrition because the cultivation is not adequate for the bees, potential immune suppressing stress. In this list, they have forgotten other insects killing the bees, and they have forgotten the climate change. But this also exists on the web, you know. Try to farm a case about climate change and colony collapse disorder. So the result is that there is a very complex situation where a lot of causes can cause the CCD. And this is exactly what the pesticide producers want. The pesticides are here with nice colored flowers here. And but they are among a series of things, exactly like for the tobacco people. The goal was not to say that there was no effect of cigarettes on lung cancer, but to say that there are lots of effects and that it's multifactorial, so that cigarette is one of the causes among others. That's exactly what is being done for pesticides. It is proposed that there are a lot of causes, and actually most of us scientists are convinced easily by that. I must admit that myself, I have been trapped in this. Because it's true that it's multifactorial, obviously. And when some people tell you that more research is needed, then you're relatively happy as a scientist. But lung cancer is also multifactorial. But in the 60s, there was one factor which was much more important than others, and that was cigarette, and it's still true. But for 50 years, the companies were able to hide the reality for tobacco by making this smoke system of other causes. This is what is going on now. Certainly, and the goal of my talk is to show you that if we see the things like that, it's partly true, but partly wrong. It's wrong because probably the most important problem is pesticides, like smoke. Cigarettes was the most important problem for lung cancer, and that for biodiversity, the fact that the others are seen as important as pesticides is an effect of merchants of doubt. Why? Well, the point is, biodiversity has been declining very rapidly for the last 30 years, particularly for insects. What has been dramatically changed for the 30 last years? What's been changed is that instead of putting pesticides on fields when there is an attack, the pesticides are put a priori on the seeds. You see, these are seeds of wheat or corn, which have been treated, and this is beta, so sugar beet. The pesticides are around the seeds. There's no way you can see these seeds without pesticides. Pesticides are poured every year on every field of grape cultures. And when the products coming from these sites fly to a hive, this is the result. The Guardian has shown that there has been a very interesting research which has been made in England. A group, a university, wanted to fund research on why do bees appear, and they accepted that Syngenta give 10% of the money of this fund, but Syngenta, by only giving 10% of the money, was able to make that the research about why do bees disappear included every kind of reason except the pesticides. This is a very interesting way of cheating with the publication system. If if you fund all research on all causes except the one you don't want to be seen, any meta-analysis will find that there are lots of causes, and that your cause, the one you are trying to hide, will remain hidden in the mass of publications. I am sure that the entomologists of America love insects, and I'm sure that they do not want to help merchants of doubt in this question. But you see, this is the site of the American Entomological Association, and what they say is that the most interesting is how much is still unknown. So themselves, they fall in the trap. But guess who is funding the American Society of Entomology? If you go to their site, you will see that it's Bayer, Corteva, BASF, Johnson, Syngenta. So the first thing is favor research in showing results which can hide the result you want to hide. The second thing is give money to NGOs which can give the results you want. The third thing which do merchants of doubt is to attack people who find results which are not the ones they are wishing. Here, a paper by Pierre-Henri showing that pesticides kill bees. This paper was published, and it was a very good paper, but unfortunately, very rapidly, there was a comment saying that the treatment was wrong and that the paper was wrong. This comment was wrong, and the response came and showed that the comment was wrong. But two things I want you to note. Helen Thompson, who is an author of the response, was at that time a member of a committee, but is now hired by Syngenta. and in the press, you know, what the Reuters gave from this story is mainly the fact that the response said that the thing was wrong. I want to finish this by showing you how incredible the situation is. In Europe, the pesticide regulation is made mainly by an organism called the EFSA. The EFSA is the organism which says whether if a pesticide can be used or not, and they have decided that in order to be accepted, to be approved, it had either have a negligible exposure to honeybees or have no unacceptable acute or chronic effects. The exposure is very difficult to measure because most of the compounds which are used today are active at the nanogram, that is sometimes under the level of detection. The acute effects are tested by the companies, so you don't know how reliable they are, but at least they are tested. But the chronic effects have not been tested until now. That is, you know, with the system we use now, we would say that tobacco has no bad effects for humans, because if you can smoke 2 packs of cigarettes in a day, you will not die. And so if the chronic effects are not known, then there is a very big gap. And the EFSA noticed that and proposed a new system to test the chronic effects of the compounds. That was in 2012. In 2012, the EFSA developed a system. And this system has never been applied at the European level. And it's never been applied. Why? Because the agrochemical industry has refused it, and it has refused it. And the incredible thing is that they have published that in a paper published in 2018. They show that if the EFSA test on the bees was used, then 79% of herbicides, 75% of fungicides, and 92% of insecticides would be rejected among those which are used today. So actually, 80% of all their compounds would be rejected by the EFSA test. It is not a paper by, I show you the paper, I'm sorry. It's not a paper by environmentalists. It is people from Bayer, from Dow Chemical, from Syngenta, who write this paper saying that 21% only of their herbicides would be accepted with the EFSA system, 25% of their fungicide and 8% of the insecticide. What this means is that these molecules, which are designed actually to kill living organisms, they kill everybody. If a herbicide, so you wonder why is biodiversity collapsing? Biodiversity is collapsing because we are pouring tons of insecticide, of pesticides every day in nature. It's been found that 75% of the biomass of insects have declined over the last 30 years in Europe, and it's exactly the same time when these pesticides have been used now. Now, you have to understand one point. If a herbicide which is designed to kill plants, or a fungicide which is designed to kill fungi, kills bees, then it's likely that it kills every animal. So actually, we are putting an enormous quantity of molecules which main effect is to kill every living organism on Earth. And if we have not understood that this is why biodiversity is collapsing, and we have not understood it, we know that biodiversity is collapsing. The IBES, for instance, is doing very nice reports about that, but the pesticides remain hidden in their discussions. The point is now, there is a new science, the science of the making of ignorance, which has been created by the sociologists and science historians I talked to you before. The agnotology is a very interesting science, showing how some scientists lead with companies, create ignorance in the fields which they wish you to be ignorant. If you want to understand the decrease of biodiversity now, you have not only to do biology, but also to do agnotology to understand why you are cheated, why you do not understand what's going on, why there are systems which prevent you from understanding, seeing, and hearing what is going on. I think that the collapse of biodiversity is a very important problem, and I think that we cannot understand it if we do not understand how the research system is cheated by these merchants of doubt. Thank you.
Thank you so much, Professor Gouyon, for this very stimulating contribution. Yeah, I think it's worth of questions. I would have several comments, but I would like to know if from the audience there is some questions, some comments. Yeah, Professor Ving, please.
Well, thank you very much for this stimulating talk. Indeed, we are living in a very complicated world, and you never know which information is good or not. Usually we scientists believe, as you say, well, what is published is good science. But as you mentioned before, apparently even our science is corrupted by people who give you money. So this is extremely complicated, especially in environmental issues. You have NGOs for something or against something. And so it's, well, the world becomes more complicated because of these controversies and this deliberately wrong information which we get. So this is apparently a problem of the modern world with social media and everything. It has, so especially with environment. So it's, as you correctly mentioned, it's very difficult to find out what's true and what's not true. But with pesticides, I think it's clear for biologists what's true. But still, in the public, it's always a different situation. And the media, they convey a completely different story. Thank you for your contribution.
Thank you very much. I would like to add that you say that for scientists, it's clear that pesticides are negative. That's certain. But the fact that they are the major cause, that they are largely over all others, is not obvious at all in the scientific community. If you look at the IBES report, for instance, they list the causes, and in the causes there is agriculture, but like a soil change system. And actually, pesticides do not appear clearly as the first problem. And I think that even in the scientific community, this is not very clear. And I think that actually, to understand what is right and what is wrong in what is published now, we really need the help of sociologists doing the agnotology work. Thank you.
And you're completely right. But even like the IPBES or whatever, big commissions we have, they are not objective. They are also driven by some sources, unfortunately.
Right.
So even if the WHO or whatever you have, you cannot believe it just as it is. Also the IPBES, some good information, but some is completely corrupted. That's my impression. So I think it's important to realize this in our world, that don't trust, be skeptic. Thank you.
Thank you.
Thank you all for this stimulating discussion. We have a question by Dr. Andrianto. I'm going to summarize a little bit. He's from Indonesia. Say, today there is a shift to use mild or low tar and nicotine tobacco in society. Do you think that will give better effect to the environment?
Well, it depends on what you call environment. Concerning the health of humans, I think that there is no good tobacco. And low tar does not change much. Concerning the environment in itself, concerning how the plants and animals survive, I think that tobacco is actually quite good because it kills humans, which are the main problem for other species. But really, if you want to improve the health of people in your country, like in any other, you have to fight against any kind of tobacco consumption. And this is exactly what the merchants of doubt have tried to hide. At the time, in the 50s and 60s and 70s and 80s, they promoted research on all other causes of lung cancer, like radioactivity from stones, genetic stuff, and lots of things. Some of them were really completely invented, some of them were real, but actually at least it maintained an ambiguity on the important causes.
Thank you indeed for this strong contribution, Professor Gouyon. I think we can move to the next speaker, Dr. Matjaž Gregorič. He's going to show us his contribution about the spider webs as the eDNA tool for biodiversity assessment or life domains. Thank you, Matjaž. The floor is yours.
Thank you for inviting me. I'm an arachnologist from Slovenia, and this presentation today is about environmental DNA that combines arachnology and molecular biology expertise. So what is environmental DNA, or in short, eDNA? This is either the genetic material that we obtain directly from environmental samples in the absence of biological source material, so in the absence of organisms, or this term is also applied to mass sampling and DNA extraction of microorganisms. And you can imagine several tissue types from live organisms are potential sources of eDNA, and so are parts and remains of dead organisms. Logically, eDNA is present in all media, like soil, sediment, water, and even permafrost. And depending on this media, depending on the medium where we obtain it from, the preservation of eDNA varies from days or weeks to even thousands of years in extremes. And when we obtain and amplify eDNA, we can do it in a single species approach, so targeting one specific taxon, or in a multi-species approach, for example, through DNA metabarcoding. Why is all this important? Because traditional biodiversity monitoring has well-known shortcomings. For example, traditional sampling techniques are many times invasive. They're mostly manual and time-consuming and highly dependent on taxonomic expertise, which is in decline or even often lacking, at least locally. And consequently, traditional approaches are often not enough to tackle modern challenges, such as the rapid decline of biodiversity, climate change, invasive species, and so on. Novel molecular approaches, such as sampling of eDNA, can potentially overcome or complement many of these limitations of traditional techniques. And accordingly, eDNA is increasingly used to address fundamental questions in many basic and applied research fields. So how do spider webs fit in here? A few years ago, a study of Xu and colleagues introduced the concept of spider webs containing DNA of organisms. Here they investigated cobwebs of black widows and obtained both spider and prey DNA from these webs. And then shortly after, another study confirmed this with amplifying spider DNA from 2 other species. Recently, a study aimed at the optimization of meta-barcoding protocols in diet analysis used spider webs among several eDNA sources of predators. And they showed that spider webs can also be used to trace organism groups through DNA meta-barcoding. However, these early studies have some limitations, and we believe that the utility of eDNA from spider webs far surpasses simple identification of spiders and their prey, so surpasses diet analysis. But why are spider webs and how could they be useful? We hypothesize that they are, in fact, something like passive air filters that accumulate genetic material from all sorts of organisms, from arthropods, plant pollen and other plant parts, fungal spores, to even microbes. And why do we think so? Why do we think they are a useful source? Because spider webs are highly diverse in architecture, size, and microhabitat. They range from centimeters to meters in size. Some have glue-coated silk, some have only bare silk. They're everywhere. They're abundant. They can be easily collected. Some are suspended for only hours or a day. Others are suspended for weeks or even months. So this can give us temporal and spatial information. And as potentially passive filters of the air column, eDNA from spider webs might provide an opportunity to address a variety of questions related to the study of communities in general, without the need to actually collect individuals from these communities. For example, I don't know, studying distributions and compositions of all sorts of organisms, tracking invasive species, fungal pathogens, and so on. The previously mentioned studies are nice in showing the concept, but were not optimally controlled and mostly conducted in the laboratory, apart from one. So to move towards a methodology that utilizes spider webs as a source of eDNA, we need to advance our understanding of 2 critical aspects. And these were our 2 goals here. The first one revolves around a single species detection in a web. So this is achieved by testing the performance of a species-specific detection of eDNA from spider webs and includes appropriate DNA isolation and amplification controls. The second aspect is whether webs as aerial filters potentially can be used to identify multiple organisms. And given that we arachnologists know that temporal and spatial factors affect spider web biology, we expect that comparative meta-barcoding should yield sufficient variation in species composition, perhaps also abundances, to allow for biodiversity estimations. So for goal 1, as in previously mentioned studies, we fed spiders with house crickets, Acheta domestica, and targeted their DNA from webs. To do that, we designed 2 CO1 markers specifically designed to target this one species. And we used 2 types of webs from spiders in our lab to test these primers. We used the Mediterranean black widow, who builds cobwebs, and the hermit spider, which builds sticky ore webs. And the idea was simply that maybe we would see differences in eDNA accumulation because orb webs are covered with glue and cobwebs are not. Then we chose real-time PCR over the previously used conventional PCR because of easier and more precise detection. And we conducted a bunch of rigorous protocol controls to be really sure that positive samples are positive and negative are negative. And we also employed a bunch of sample controls, for example, to control for the specificity of primers and for potential inhibitors of DNA amplification that could potentially be in spider silk. Our primer results and all the controls were as predicted. The primers worked well across diverse DNA concentrations. And one primer set consistently performed better than the other, so we used this one in subsequent work. So then we went to the real goal. We went into nature and we tested for prey detection from webs in nature. And we chose webs of two cohabiting spider species, the garden spider Araneus diadematus here in the photo on top, and the common hammock weaver Linyphia triangularis in the bottom. So the garden spider builds sticky orb webs and the hammock weaver builds non-sticky sheet webs. We threw two tiny juvenile crickets, only a few milligrams in mass, into each web, and we used 5 webs of each species, and then additional 5 webs as control. We successfully traced house crickets in all 10 webs, and we also successfully traced house crickets in all samples when we diluted them 10-fold. And this was kind of in the threshold of detection, but these diluted samples are not shown here on graphs. I do mention it because this means that even tiny amounts of cricket DNA could be traced. At the same time, all negative controls were negative and all the internal isolation controls were positive in all samples. So this, in other words means that the PCRs worked and these are real prey detections. As said, for the second goal was to test whether, like through a eDNA metabarcoding study, whether eDNA from spider webs yields sufficient variation in species composition and abundances that we could use this information for biodiversity estimations. Again, we used the same 2 spider species, the garden spider and the hammock weaver, and we sampled these webs in 2 forests, one continental, the other sub-Mediterranean. And for one of these forests, we sampled in 2 subsequent years. Each time, again, we sampled 5 webs per species per forest, and each web was its own sample. And the idea was very simple. It was not to target only spiders and their prey, but to try to amplify everything we can of a certain organism group. And we hypothesized that we will detect differences between web types, forest types, and sampling years within a forest. First, we tested a bunch of markers for several organism groups, and all these markers have been shown to perform well in past metabarcoding studies in all sorts of environments, from water to land. They mostly work quite well. We had some problems with plant primers. So then we finally decided and chose 3 markers targeting animals, fungi, and bacteria. We performed high throughput sequencing of the amplicons using the Illumina MySeq sequencing platform, and we analyzed the data using the QIIME2 software package. And the databases to BLAST2 that we used were Silva for bacteria, Unite ITS for fungi, and BOLD for animals. To render all these samples comparable for diversity analysis, we randomly sampled reads from each sample and in that way equalized their size. And when we were deciding how many reads versus samples to retain, we maximized the number of samples rather than the number of reads. So from the 25 webs in total, we retrieved at least in my opinion, a surprisingly high amount of SAVs, so amplicon variants. Bacteria predictably represented the largest diversity, followed by fungi and animals. And here in orange, you can see a simple overview in some simple numbers to get the idea of the diversity of taxa in these samples. And this really surprised me. I was in between a little bit afraid I would get many blanks since single webs were samples. The analysis of alpha diversity showed that in the two forests and across years, all webs together accumulate a similar diversity of bacterial, fungal, and animal DNA. However, sheet webs accumulate a higher diversity of bacterial and fungal, but not animal eDNA. And here I have to note that the sticky orb webs are suspended only for a single day in this species, while sheet webs are long-lasting while they are not sticky. So maybe this is a little bit expected. But more interesting, while alpha diversity only tells us something about the number of taxa in samples, beta diversity analysis compares compositions. So even though alpha diversity was similar, the species compositions within these diversities were not. And our analysis showed that sampling locality, web type, and sampling time all affected the inferred community compositions of all three organism groups. These PCI plots here visualize these differences in community compositions. They're a little bit difficult to read, but I hope it gets the message across. I also want to note that we used both indices that use presence-absence information only, as well as those incorporating abundance data. And all these indices show similar results, but there's too many to all show here. In other words, though, while we show that many organisms leave genetic traces on webs, these genetic traces seem to give enough resolution to distinguish among localities, between sampling time and sampling type, if we think of different web types as sampling types. So to conclude, we argue that spider webs prove suitable for detecting genetic traces of organisms in both a targeted search for a specific species and within a meta-barcoding approach, because we show that the targeted search for eDNA from individuals of a single species is a valid concept. And we also show that spider webs do seem to act as passive filters of the air column and are a promising tool for general biodiversity monitoring through DNA meta-barcoding. But of course, to get to very precise protocols, additional targeted work on single groups is needed. And indeed, our future plans include a step towards establishing such protocols for monitoring some arthropods, including pollinators and invasive mosquitoes, and even airborne microbes. And we also want to test some very spider-specific, web-specific hypotheses using eDNA. Thank you very much for your attention.
Thank you, Matjaž, for this very nice presentation. I'm really impressed about the considerable amount of biodiversity you can catch with such a techniques that couple nature with new technical approaches such as meta-barcoding. My congratulations.
Thank you.
I have some some question, but I leave the floor to the audience. Here we have a question by Anatoly Kapugi, I guess. Again, I'm going to summarize a little bit. He's saying, do you have plans to involve data from outside of Central Europe in future?
Not currently, because of course it's much easier to conduct research in your backyard or a forest or meadow close by. So, and you have to combine a lot of expertise to test this method. For example, for pollinators, we are doing a parallel study with classical methods, insect traps, and then we need taxonomists who determine all these bees. So we're doing that at home now. We do think of in the future of perhaps doing like a pilot study in Madagascar, but we'll see about that. There's no very specific plans for now.
Okay, he's also saying that you wonder if you have some public proposals, but I guess you have already answered about that, which could be used by specialists to send data or to your teams. Yes. Or even citizen science is another.
Yeah, these are also protocols that are quite simple, and the molecular methods are well established and can be done in practically any molecular lab in the world. So anybody can take this idea and use it at home. So they don't necessarily need to collaborate with us, and unless they know they need arachnological expertise, I would say.
Okay, thank you. Another comment by Sarah Fisher. I'm going to read it. Great talk, Matjaž. This research has great potential all over the world. We have big orb spiders here in Australia, and also spiders that use a ground web, such as trapdoor spiders. Any thought to looking at ground dwelling spider webs? This is the question by Sarah.
Yeah, one goal of this study was to use two very starkly different web types. And both are single species, but many spider species build webs. And these webs are even within a type, within orb webs or sheet webs or cobwebs, can be suspended in many microhabitats. So depending on what one wants to answer, what kind of a question one wants to address, yeah, why not sampling ground spiders? But, so for example, for pollinators, we didn't. We sampled other types of webs, but I'm pretty sure there are some research questions that somebody can think of and sample ground spiders.
Thank you so much, Matjaž. Any?
It's me, Michael.
Yeah, Professor, please.
Okay. Nice talk. So one problem with eDNA is you need a good database. That means all the sequences of the species you're looking at must be deposited in a database. It's true for vertebrates, but I'm pretty sure for a lot of insects, you don't have reference sequences. So my question for you is, with all your reads, how often could you go down to the species level, and how often did you fail? So only maybe family level.
To get to the species level in animals, it's not so common. And even when I did get to species level, many times I couldn't be really sure that it's the correct species, but I could be fairly sure that it's that genus. For this study that is more about the proof of concept, I wasn't so much worried about that. It's true that for invertebrates, the databases are quite poor, so I had to do a lot of manual corrections and checking for blast results. But in the end, what I wanted to really know is if I can get any resolution between localities, years, and so on. For the future work, I mentioned pollinators. We're actually combining what is already published to our own sequences that we collect in parallel. So the pollinators collected in classical insect traps are all being barcoded, and we will combine our new barcode data with existing barcodes to be as sure as possible that what we will get in meta-barcoding are appropriate results.
Good. I think it's an important limitation. For the ecologists, that's fine. But if you go for taxonomy, they always want to have the species or even finer resolution. So even at the moment, e-barcoding, as you correctly mentioned, very often fails to identify the species. So at least one should not be too optimistic to get a real inventory of the species present in an area, but you can do nice ecology, as you pointed out. But it will change because a lot of more sequences are being generated. So I think in 5 or 10 years, we can really go down to the species level. What do you think?
Yes, it doesn't necessarily mean that such a molecular approach can completely replace everything that's been, all the methods that are in place so far, but we can certainly complement these methods, or for certain questions, we can do it easier and more cheaper. And time will tell, of course, about all the uses of such methods.
And in this regard, a question and comment by Geraldo Freire, and congratulations for your talk. But what will be the challenges of this method in tropical areas with a huge biodiversity? I think this could be a field of application very, very interesting. What about?
I'm not sure what the limitations are. I guess we'll see when we do it, but from the methodological point of view, I don't think there are many limitations, because the sequencing technology improves very rapidly, and we get such insane number of reads back that I don't think that the local diversity will present a big problem, honestly.
Yeah, it was in a positive vision. I mean, in areas with such a great biodiversity that could be hardly manageable with, for instance, with traditional methods. Just as you pointed out before, a complement to the classic approach could be of help in this case. So, Matjaž, many thanks for this great talk.
Thank you for the invitation.
Okay, I'm going to introduce Dr. Wilson Frantine-Silva, who is going to give a talk about a striking frequency of diploid males in a less suitable environment, the future of the orchid bee, Euglossa annectans. Please, Wilson, the floor is yours.
Okay, thank you. Thank you, Professor Paolo. Thank you for all this stuff, for having me. So, okay, as Professor Paolo said, my name is Wilson Frantine-Silva. I'm a PhD at the State University of Northern Rio de Janeiro, in Rio de Janeiro, Brazil. And I'm very glad to have this opportunity to talk a little bit about this very interesting case about such amazing bees, which is this striking diploid male frequency. I will show you a couple more in the next few slides. Of course, I'm here on behalf of my colleagues, Professor Dr. Maria Cristina Gregorič and Professor Dr. Silvia Sofia from the State University of Londrina. This is my email, so if I have any questions after the presentation, they will be very welcome. So, as Professor Gouyon just said before, we have like this problem with the bees, not just with bees, but bees have been tracking for a couple times. So more than just the colonies of the honeybee, we have problems with the whole diversity, right? This is a kind of a brand new data from the last month. I think it's published in February, if I'm not right, or not wrong. We see in this worldwide data set that we have tracking records for bees from the last century, and we see that even when we got like more records, we got few like 25 less species than we got 3 years ago. So no matter if you're looking at the whole records, which include the Pesticides, or if you are looking at the only museum species, we got this clear trend to lose species. So it's a clear indication that bees are really declining. So of course, we always ask about what are the main drivers behind this bees decline, right? We have, of course, for any other kind of animal, we get the same pattern, which is probably the habitat loss combined with pesticides, particular for insects and bees, of course, and climate change as well, right? We have this very elegant example with the North American and European bumblebees that shows clearly that the north limit of the distribution has remained somehow stable, and the southern limit of distribution has clearly declining through time. So when we compare the data from the last century and the first decade of the 20th century, so this is a very clear evidence that the climate change is shifting species distribution, right? So here in Latin America, we have just an additional issue, which is about what we don't know about those bees. We actually don't know how much we have lost, right? This is also a very brand new data. When we see, again, global biodiversity information facility data, right? And we see that in a ring, how much from where this records came from and how many species they represent. So as we see, we have 26% of the 20,000 species, but we have just 7% of the records. So we really don't know how much we have to lose or we have lost. So, and here in this map is just a distribution of those records. So we are really, really actually lost in this situation, right? In the middle of this 20,000 bee species, we have this very beautiful group of bees, which are the orchid bees. They compose a tribe of apid family, which is Euglossa. They are worldwide known by collecting perfumes from different sources. Here in this picture, we have males collecting these scents to present to females for the partnership. And of course, they have evolved to present this kind of a behavior. And orchids has evolved as well to produce this kind of scent, and sometimes make a couple traps to make the bees like act as pollinator for this species. We have some species of orchids has this kind of a syndromes. We have documented something like 700 orchids, which are exclusively dependent of orchid bees for pollination. This group has about 2,500 of species in 5 genera. Here we have pictures of those genera, like Euglossa, Eulema, Euphrisia, Ixeret, and Agli. And they are, of course, no tropical bees, as we can see here. Their natural distribution, we have records of one species, invasive species, in this case at Florida, and one species in Jamaica, if I'm not wrong. They are known as pollinator of several angiosperma families, and very important pollinators actually in the rainforests. The focus of this studies is the Euglossa annectans, which is one of those species. It's endemic to Atlantic forest, and we know that pollinates more than 24 families of Angiosperma. And we already know that it presents a high rate of diploid male. I will show you that it can be a problem for a bee species, right? Because in a general way, when considering loss of biodiversity, we generally considering loss of the taxa itself. And we not often discussed about this another layer of diversity, which is the genetic diversity, right? In the case of the bees, it has more even concerning, because as I'll show you, bees have this kind of haplodiploid, just like any other hymenoptera, this haplodiploid sex determination system, in which the females are diploids and male are haploids, right? And that's all depends on this complementary sex determination gene, or loci, which depends on heterozygosity to trigger the cascade to generate a female, right? So if you got two different alleles, just like here, AE, AI, and AJ, its product interacts with the feminizer gene, which will transform this to trigger the cascade for the females. When we have just one allele, it skips the female step and feminizer step and go directly to DSX to trigger a male. However, when we got less genetic diversity than necessary, we start to get this association between two equal alleles, which will work exactly how the one allele in haploid males, right? So it will skip the feminizer step and will give rise for a male, but a diploid male, which is often sterile or it's not able to mate, right? It's awful for the population, because we start to get into this diploid extinction male vortex, which is a decrease in population size, or because external pressures, just like pesticides or climate change or whatever, it will decrease, of course, the CSD richness, which will increase the diploid male production, which will decrease the population growth rates and will decrease population size, and we'll get this vortex until the extinction. Of course, these organisms, they have adapted some mechanism to escape actually such a kind of situations. Otherwise, hymenoptera won't be so diversified as they are today. But it is still a concern when we got small and closed populations. So that brings us to our question, which is very simple, actually, we are asking whether Euglossa annectans is really in trouble or not, right? So we break this question in two sub-questions, which is one of the main concern. How the first one is how climate change has affected or will affect its distribution. So the first question of this study. To address this question, we have built some ecological condition models. I won't go any further with this explanation actually. And we just did in the Maxent, and with 3 GCM and 3 variable selection strategies from 71 records mined from the literature and some museums we had access. We then got those results. We projected these results to the present, to the last glacial maximum, and to the future for an average scenario. The RCP 4.5, it's considered as moderate, not exactly nor optimist nor either pessimist. So from each one of those projections, we got binary maps, and we calculate the predicted distribution size as counting of pixels, the area corrected to square kilometers, and the core area, which is the size less the perimeter, right? And of course, to quantify the diploid male production, to see whether it is a real threat or not, we then genotyped 398 males from 12 localities for 8 microsatellite loci, right? We then just count the diploid males and quantify the diploid male productions. And we, of course, quantify some genetic diversity indexes. There is just a slide about how ecological niche model works. I won't go much deeper in that because Professor Lucia already talked about this. But just to keep in mind that we have this sampling points, we got this predictor variables, we impute that in some algorithm, in our case the maxent, and we estimate then the niche or this virtual niche space, and we then project and validate this model to project this n space and time to evaluate, for example, if that particular space is suitable or not for invasive species or whatever. Or we can also access the shift, the range shift through time. So just going here to our results, we got this 71 verified records from the literature and review collections. It's just to give you guys a picture on how it is distributed. We have here just a frame of the Brazilian coast, the southern part. Have the yellow dots, which are the records from the literature, and the green squares with numbers, which are the sampling sites where we got the genetic samples. Okay. Then directly the results of the niche models, what they have tell us. They have tell us that the suitable areas have changed considerably since the last glacial maximum at 21,000 years ago. And this suitability has decreasing over time. Right here, the red areas are the more suitable, and they are decreasing according to the scale. And as we can see, we have this those two big areas in here, one just right at the coast, where is the continental platform. Now it's under the sea, actually. And this in-country area, which has become just one here in the present with very important decreasing. And we can also see just visually in here that we keep this trend in time, right? For decreasing. In binary maps, we can quantify this as percentage from the shift, right? Here we can see that the distribution area decreased 42% at the present when compared to the last glacial maximum, and decrease again to last 36% when we project it to an average prediction for the future, right? So if we're considering that things are going well and not worse, we go this very important decreasement in the suitable area. Things got a little bit worse when we got this, when we analyzed the Atlantic forest, the rainforest that left, right? We have this very highly fragmented scenario in here, and we have this yellow area, which are the prediction of the model. This line right here, which is the limits of the Atlantic forest, right? And this green, like this green spot, like like dust in the map are the fragments. And we see that we have an important decreasement at the west part. These other areas highlighted here are protected areas, and we are losing important suitable areas in areas that should be under protection, right? This is just the numbers that we estimated from where we estimated the percentage. Here we see the difference, for example, in the area. It decreased importantly when we considering just the comparisons between LGM, the current time and future. And of course, when we consider the current patches and future patches, we have this, which are the scenarios considering the Atlantic forest that left, right? That we have left. And another interesting stuff to considering is that we have seen some studies pointing that orchid bees are likely to reduce abundance even in the climate suitable areas. So when we think about that, we think about the space reduction and also the abundance reduction. We go back to the diploid male frequency, right? Euglossa Annectans do have a outstanding diploid male frequency, right? We can see here almost 10% of diploid males on the whole data set, just like considering the whole males genotype. And when we compare it to the other orchid bees, we see that we have this huge difference on the estimation, right? We'll also see that higher frequencies not always means high numbers of diploid males, because we have here outstanding frequencies, but just a few of males because it's based on small sample sizes, right? So that's important to look at a bit more large samples. But it is still a lot when we compare the 39 diploid males against this total of 398. When we compare that with the other studies, which found just 5 diploid males in 1,010 males genotyped in 27 species, actually. So that's quite a lot. The conclusions here are quite predictable, right? We have this very remarkable retraction of the suitable areas from Western, and we have this outstanding diploid male frequency confirmed. So I'm skipping just some points in here, but we at the present situation we still have possibilities to management to keep viable populations despite of all those diploid male frequencies. However, the fragmented scenario is quite concerned. The good news is that the suitable areas are shifting toward the part of the forest that we have the largest fragments. So that's a good news actually. But in the present scenario, when Brazilian agriculture areas are getting bigger and bigger, we have like a very concerning scenario about this species. I just would like to thank some sponsors of this research, which is CAPS and PK for Pierre-Henri, and also the environmental organizations that have helped us through the sampling expeditions. So this is it. Thank you.
Thank you so much, Wilson, for this interesting contribution. We already have a question for you by Dr. Capughi. He is asking, did you consider such extinction driver as extinction of orchids or non-orchids plant species pollinated by the study bees?
Yeah, okay. So that's an interesting question. But as far as we know, orchid bees has evolved not so dependent from orchids. Orchids are just a little bit more dependent than from orchid bees than otherwise. Of course, there are a couple of pair of species which depend from each other, but the most part of time, orchid bees do well with dialogue orchids because they can collect scents from other sources when available.
Okay, thank you. This is it's a common problem for instance, for obligated epiphytes that need their substrate. It's the story for my beloved lichens. If the tree is going to disappear, of course, the obligate epiphyte has no way to survive. So it's again a story of a relationship. But in this case, we have some hopes for the bees.
Yes, we do.
Okay, any other questions? Yeah, by Manuela Giovanetti. She's going to ask you, what about the bees' behavior? Is it similarly influenced, I guess, by the global change, well, by the drivers?
Oh yeah, no, about the behaviors, we don't have any evidence that tells us that the behavior are changing. But we just have we don't have actually much research on that. From the field observations, we just see that things are doing well. There's a professor in Panama which works with these bees for like 30 years, and he just said that they're doing well at that part of the world. So it's not exactly the same for down here in Brazil, in this most developed part of the country. So I don't know. It's for this specific kind of behavior. As far as we know, they are doing well at this point.
Yeah, and Manuela is adding a remark. Since changing in mating behavior can influence your data, the rate of diploid male.
If the
suggestions and with
yeah, no, I don't think so. As I said, the behavior for mating, they still like quite similar. Actually, the diploid male, they act exactly the same, the normal males. So we just know they are diploid male when we genotype them. They go to the scent baits exactly like normal males, and we are not so sure that if they can't mate or if they mate, they will they won't be viable, like if offspring actually will be viable. So I think they it won't be like any flag at this point, I think.
Okay, thank you so much.
Thank you for having
Manuela is commenting, yeah, perfect, that was my idea. Maybe we need more data on that, and thank you.
Thank you.
My thank you also for you, Wilson. And now we are approaching to the last contribution by Dr. Maria Panitsa. So, if I'm not wrong, we are at the end of this nice online session of the first Biological Diversity, Ecology and Evolution online conference. Just some final remarks. I thank you so much, the speakers, for their contribution that gave us a range of situations, ranging from from plants to animals. So covering on several groups of organisms, covering several levels of biodiversity, starting from the species level, coming to the genetic diversity, as mentioned correctly by Wilson. And again, what I think is emerging from the session of today is that we have we have problems, and maybe even some still positive situation, as reported by Maria in her final presentation. We have problems because it's not a secret, but it is already well evident that we are going to have serious critical problems for biodiversity in the following years. Or even worse, we are already in problems. We are already in troubles, and what is expected is that we actually don't know if we are still in time to find solution. But on the other hand, this should be taken as a challenge for scientists, as we are, to try to do our best for saving what we could still save. And for instance, Lucia showed us that we will have Lucia Varaldo with her presentation, that we will have the possibility of adding new techniques such as translocation techniques based on an improved knowledge on which is built on species distribution models. So coupling a statistical method with active conservation practices. And well, and again, as our colleague Matjaž Gregorič told us, we are still discovering new methods even when we are with approach we will never thought about that, with the for instance coupling the observation in nature of in this case of spider webs with new technological approaches such as the eDNA. And again, Wilson informed us how we should go a bit more in deepness, focusing the attention not only on the species levels, but taking into account what is going to happen to populations, for instance, and taking into account that several species have a characteristic biology. For instance, in the case of bees, as he informed us, the issue of the diploid male could be an additional problem for the conservation of these species in the near future. I'm forgetting. Well, Maria told us something good, basically, that she is informing us that notwithstanding with the critical situation we are experiencing in the world, we still have a lot of great biodiversity hotspots that are currently experiencing a speciation process. So the diversity is still with us, what we can say. I'm taking last but not least the contribution by Professor Gouyon, who warned us about an additional risk. So we are experiencing a decreasing of biodiversity, but we also have to face another risk, which is not less dramatic or less risky. And this is the risk of facing the false, basically, right? So the new challenge is the struggle for the truth. And the truth in science is providing scientific data, robust scientific data based on a scientific approach. And as Professor Gouyon told us, it's not always so simple to to fight the false information with scientific methods, because well, we are used to approach with an experimental way of thinking, but this is not always the case, that this is sufficient for informing the population about what is true and what is not. So another challenge, if I can summarize this late aspect, is to find the border where the scientists have to work. Is it sufficient that biologists, ecologists, and only do their scientific work, or they need to involve a little bit more on the scientific debates with the public? Okay, that's all. I hope to have summarized the main points, but of course the discussion is still open for a few minutes if you want to add something, or otherwise I thank you for your attention. Thank you all the speakers for participating in this nice and in some cases strong discussion about biodiversity and conservation. So Charlotte, I leave the floor to you, and thank you for the assistance and for being here.
Thank you very much. You kind of said it all. So thank you for chairing this session. Thank you for the speakers, and Professor Wink as chair. Thank you for your questions. There are still 2 more live sessions for this conference held next week. I remind everyone that registrations are still open, so don't hesitate to register for these live sessions. Wishing you a nice week and hope to see you at the next live session.
May I say a few words?
Sure.
Because as the chairman of the conference, I'm very happy about this day. Many thanks for Paolo. You've done a good job. You organized the session extremely well. Thanks for the speakers. I heard interesting talks, and I'm really happy that we have this diversity of experienced scientists, of young scientists, of interesting topics. So this was more or less the idea of this e-conference. So it's a new format, and it seems to work. Also thanks to MDPA for Charlotte to organize it. Well, thanks for everybody. Thanks for listening. And again, for Paolo, fantastic. Thank you very much. Bye-bye. See you next week.
Thank you, Professor.
Bye, everyone.
Thank you.
Bye.